After we swallow our food, it will then be passed along the GI tract. Now, while there is skeletal muscle in the oral cavity and along the upper portion of the esophagus, this very quickly then becomes smooth muscle. And from this point on, then involuntary reflexes, often governed by the E and S, the enteric nervous system, this is what will move the ingested material from this point on. Along the majority of the GI tract, there are two layers of the um, muscularis, and in the stomach there are three. There is an inner circular layer of muscle where the muscle squeezes behind the bolus, and an outer longitudinal layer of muscle where the muscles contract in front of the bolus. And you put this together, and thus instead of contraction, we don't speak of the esophagus as contracting or relaxing the way we would say the biceps. Instead, we talk about peristalsis, this wave of muscular contraction, which passes along the esophagus, in which the longitudinal muscle in front of the bolus contracts and the circular muscle behind the bolus contracts. Peristaltic waves then also pass over the stomach, the small and large intestines, and then even other organs such as the oviduct or the vas deferens or the ureters. So peristalsis involves these waves of muscle contraction using these two layers of muscle. Then the ingested material will enter the stomach. And as the chapter focuses on anatomy, primarily we begin with the anatomy of the stomach. It is a J-shaped organ with an outer greater curvature, a smaller, lesser curvature from which the greater and lesser omentum originate. There are three layers, as I had said, and we can split the stomach into regions. The cardia region, where the esophagus empties into the stomach, the fundus region, the majority known as the body, and then the area where it narrows to meet the small intestine, the pylorus. Just a quick note on the fundus. Typically, the fundus is not needed, but whenever a full meal is eaten, say on Thanksgiving, it is too much for the body to process. So some of the food will then be pushed into the fundus while the first portions eaten can be worked on in the body. Now, I know it seems odd. Why would we have a part of the stomach for Thanksgiving? But the way that many humans eat. So, for example, earlier humans, if a large game animal was killed, then one would want to eat as much as one could at that moment, and then leave before the cats and the wolves, et cetera, then arrived on the site. And so, eating a lot when food is available is something that humans in some situations have had to do. Now, the GI tract has sphincters, kind of like the doorways in a building. So in a building, you have a hallway, say, and a room, and this space is continuous. You can go from the hallway into the room, but then there's a door where you can close the door and thus control movement, preventing movement, which is inappropriate. In the same way here, the GI tract has muscular thickenings known as sphincters, which control movement. So essentially, this is the entrance door to the stomach. Nothing comes into the stomach until this door opens, until the sphincter relaxes to allow food to pass from the esophagus into the stomach. That's the esophageal sphincter, sometimes called the gastroesophageal sphincter. And in the same way, there is an exit from the stomach, a pyloric sphincter. So as peristaltic waves pass over the stomach, a bolus of food is pushed here. Notice that this did not originally open because this is not a race. It is not the goal to get this food through the intestines as quickly as possible. Instead, the intent is to get every single nutrient out of this ingested food. Remember, we're expensive, and the only way we're going to survive the day is if all of the calories and nutrients are extracted from this. And so rather than being pushed through quickly, rather most of this is now going to double back and be mixed again and spend several hours in the stomach. When food enters the stomach, it is a soft, squishy bolus of food, but when it leaves, it is a semi-liquid juice known as chyme. So the stomach is primarily here for mechanical digestion, to convert this food into chyme. And then when parasalted waves bring uh, the the material towards the small intestine, the pyloric sphincter will open, allowing a bit to squirt through, while the rest of it will then go back and be processed more. And so the stomach is primarily here for mixing. And along the stomach, there are different types of cells. The cells are often organized into these gastric pits, and there are different types of cells. So we have a surface epithelium, all right? So while the lumen of the stomach is acidic, these cells are producing an alkaline juice which protects us from this stomach acid. There are mucous neck cells. There are parietal cells, which are secreting the primary enzyme known as pepsinogen. It is made in its inactive form, and it is only in the acid of the stomach that pepsinogen is converted into pepsin, which then digests proteins. And then the chief cells make acid. Now, the acid is important because it kills microbes and thus helps to protect our bodies. But this acid also then activates pepsinogen and then helps to denature proteins and aids in the digestive process, making the proteins more easily to cut apart. Just a quick word on the acid. While it is hydrochloric acid in the stomach, technically that's not how it's made. So technically, the acid that the chief cells make is what's called carbonic acid. Carbon dioxide, CO2, and a hydrogen ion form carbonic acid, which then produces a hydrogen ion and bicarbonate. The hydrogen ion will go into the stomach lumen along with a negative chloride ion. So this negative chloride ion changes places with bicarbonate. So negative bicarbonate goes into the blood, and this hydrogen ion goes into the lumen of the stomach. For that reason, it's hydrochloric acid, which is in the stomach lumen. The bicarbonate, which goes into the blood when we eat, not only helps to form a blood buffer, but also one of the reasons that we get sleepy after a big meal. Finally, notice at the bottom there in green, there are what are called G cells, or enteroendocrine cells. They make the hormone gastrin, which will stimulate the stomach. So while the primary role of the GI tract is not to produce hormones, nevertheless, there are cells which produce hormones, such as these G cells here. Okay. So I have a video on the role of stomach acid, which I had mentioned. The acid does a number of, completes a number of roles. Now, not only does the stomach empty into the small intestine, but in the same region, so do some of the accessory structures. So I'd like to go through them first. The pancreas also empties into the small intestine. It is an accessory structure so that food does not pass through the pancreas, but nevertheless, its secretions are important in digestion. We can separate it into a head, the region which joins the intestines, a body, and a tail. So it narrows as it forms the tail on the left side of the body where the spleen is. While most of the pancreas is exocrine tissue, making digestive enzymes, about like this stuff over here, about 1% of the cells are also endocrine. So there are these islets of Langerhans which make insulin and glucagon. And so there's also an endocrine pancreas. Since it is mostly exocrine, then there's obviously an exocrine duct a large pancreatic duct through which enzymes will pass and empty into the small intestine, and a smaller accessory duct because when we were embryos, the pancreas had two parts which fused. So the pancreas is an accessory digestive organ with an important role in making digestive enzymes. And now to the liver. Actually, come on. There we go. The liver is the second largest organ in the body after the skin, and is separated into a number of lobes. So there is a large right lobe, there is a smaller left lobe, and then on the inferior view, one can observe a number of structures near the gallbladder. There is the quadrate lobe, and then near the inferior vena cava, there is the caudate lobe, right? And so we have 4 different lobes of the liver. The falciform ligament separates the right and left lobe and anchors the liver to the body wall. And we can even see a few depressions for where the liver contacts other organs. Right? So there we see the quadrate lobe and the caudate lobe. Now, we'll have to talk about blood vessels and bile ducts because you can see that clearly they enter the liver and are important. We could refer to this if we wanted to as the hilus region or the hilum of the liver, because notice that the hepatic artery, the hepatic portal vein, and the common bile duct, are all approaching I'm sorry, that would be the hepatic ducts going into the liver, would all enter the liver at the same point. So we could call that the hilum or the porta hepatis. So there is a hepatic artery which brings oxygenated blood to the liver. But now obviously we have to explain what that purple blood vessel was, the hepatic portal vein. Most circulatory systems in our bodies have an artery, then a capillary, then a vein, and then you go back to the heart. There are 2 places in the body where we have portal veins. So between the pituitary and the hypothalamus, you have an artery go to the hypothalamus, form a capillary bed, then a portal vein that goes to the pituitary to form a capillary bed, and then a vein that goes back to the heart. So there is a portal vein between 2 capillary beds. And the same thing happens here, although this portal vein is obviously much larger. When you consider the inferior vena cava, which will go back to the heart, all of these veins could empty into the inferior vena cava. It's right there, but they don't. So when these veins bring deoxygenated blood with remains of your last meal, they do not empty into the inferior vena cava and bring blood back to the heart. Instead, they empty into what's called the hepatic portal vein. So the veins that come out of the stomach, the intestines, the spleen, the gallbladder, they could go into circulation and go back to the heart, but instead they form this hepatic portal vein that goes to the liver. So the liver gets your food first before any other part of the body, sampling the majority of what you've eaten. The liver gets it because this hepatic portal vein will then break into capillaries in the liver. Why? Well, because the liver does so many things. It has more than 200 functions. So the liver does carbohydrate stuff. So the liver can convert monosaccharides into polysaccharides for later. It can convert some monosaccharides into others. It can convert amino acids into glucose. It can convert glucose into triglycerides. It can store glycogen. It can break glycogen down. So the liver does carbohydrate stuff. So it would like, to get your carbohydrates first. The liver does fat stuff. So the liver can convert things into fatty acids. The liver can store triglycerides. The liver can make cholesterol. It can synthesize bile salts. It can make the lipoproteins, which help us transport lipids in blood. So the liver wants to get your food because it does lipid stuff. Your liver does amino acid stuff. It can convert one amino acid into another, or amino acids into glucose or triglycerides. So your liver wants proteins because it does amino acid stuff. And then finally, your liver then protects us from harmful things in our diet. If we've ingested alcohol, then it will make alcohol dehydrogenase, which detoxifies this. It will detoxify drugs and toxins, which we ingest from plants. There are plenty of carcinogens in the plant material which we ingest. It will break down hormones so that your body won't be affected by the hormones of organisms you've ingested. It can bind heavy metals to a degree. It can perform phagocytosis of bacteria. There are white blood cells of the liver known as Kupffer cells, large macrophages. They are ingesting old, worn out red blood cells. This was mentioned in the blood chapter, so that red blood cells don't burst in circulation. Rather, we control it. So one of the places where red blood cells undergo phagocytosis are in the liver by these macrophages known as Kupffer cells. But then these can also then phagocytize foreign material. And in the next video, the Kupffer cells ingested black particles to stand out. So the particles which you see in the liver are the Kupffer cells, which under normal conditions can ingest microbes, but they can also ingest old, worn out red blood cells, or whatever the liver determines. So the liver does have these 200 functions, and that is the reason that it gets your food first. Okay. So then the liver then has two sources of blood coming to it. Not only does it have oxygenated blood coming from the hepatic artery, so the abdominal aorta gives rise to the celiac trunk, which then forms three arteries, the left gastric, the splenic, and then the hepatic artery. So the hepatic artery coming from the celiac trunk, from the abdominal aorta. This brings oxygenated blood to the liver. That's about a third of the liver's blood supply. But about 2/3 of the liver's blood supply comes through the hepatic portal vein. So the veins from the stomach, the intestines, et cetera, don't go to the inferior vena cava, but rather go through the hepatic portal vein. So then the liver gets a sample of then, what we have ingested. After passing through the liver lobule, which I'll describe in a second, then there are hepatic veins which form from the liver, and then the hepatic veins empty into the inferior vena cava. Okay, and so the flow of blood through the liver is just a little more complex, and we can see that then in the liver lobules. When you look at the liver under the microscope, you see these blocks of tissue, roughly hexagonal in shape. And so along the outside of the liver lobule, what are known as portal triads, there are holes for branches of the hepatic artery. So this is oxygenated blood. So oxygenated blood coming from the abdominal aorta reaches the liver. But then you also have the deoxygenated blood going into these branches of the hepatic portal vein, and then they mix. And so the blood flowing through the liver is then a mix. It has both the oxygenated blood from branches of the hepatic artery, and then deoxygenated blood from branches of the hepatic portal vein. These then mix to form these sinusoid capillaries. And so going throughout the liver, there are sinusoid capillaries here in the purple regions. If you remember, there are different kinds of capillaries. There are continuous capillaries, fenestrated capillaries. The capillaries with the largest spaces between cells are what are known as sinusoid capillaries. That's because big things are entering and leaving the blood at the liver. So red blood cells, can undergo phagocytosis. But also the liver is extracting things from your diet, but it's also adding blood proteins. So complement proteins, clotting factors, albumin, and plasma proteins. They end up in the blood, but they were made in the liver. So big things are entering and leaving the blood in the liver. So there are sinusoid capillaries here whose large spaces then allow for this transport. But then once again, the way that blood flows through the lobules of the liver is that we have blood coming from two sources, a branch of the hepatic artery, a branch of the hepatic portal vein. These mix to form sinusoid capillaries. This then flows into a central vein in the center of the liver lobules. Central veins then fuse to form hepatic veins, which empty into the inferior vena cava. While blood comes in from the outside and moves towards the center, bile is actually flowing through bile canaliculi to the outside of the liver lobules. So then just obviously this is just a synopsis of the material that's in these individual videos, and obviously I encourage you if you wanted to see anything in greater depth to go there. But once again, so if you were to look under the microscope, the liver is organized in these blocks of tissue known as lobules. Here in those spaces, these are all those sinusoid capillaries entering into a central, oh, I'm sorry, we're not in the central vein, my fault. So here you see what's called a portal triad, where there is a branch of the hepatic portal vein, a branch of the hepatic artery, and then a bile canaliculus. These are on the outside of the liver lobules, and then they flow into the center where the central vein is. So we would have the portal triads on the outside of a liver lobule, and then blood flows through these sinusoid capillaries before reaching the central vein. Now, sinusoid capillaries have the largest openings, so the larger materials can enter and leave the capillaries at these points. And that's because the liver is doing so many things. As I said, it's extracting nutrients, so it can store monosaccharides as glycogen. It can, do all of these things. So the liver certainly wants material to enter and leave. Here are those Kupffer cells, as I mentioned, etc. Okay. Other things, just, these are come up in other videos. The liver can store fat, but if one then ingest too much fat and in weight gain, then the liver can become too fatty. This is fatty liver disease. And then also just fascinating, I have a video on liver cells are just weird in that they are very often tetraploid, having 4 copies of each chromosome, but they can be octoploid. And thus, they have their chromosome arrangements are different from typical outside the body. And thus, liver cancer potentially arises from the liver because it has all of these abnormalities to begin with. Let me get back to bile, because bile is needed to emulsify the lipids in our diet. The liver makes bile, and then the bile canaliculi empty into what's called the hepatic ducts, into the common bile duct, which then goes into the small intestine. So the bile in the small intestine, which is, oops, sorry. I don't think this is the other one. And the bile in the small intestine was made in the liver. However, we don't always need bile to emulsify fats because we're not always ingesting fat. All right. See, bile salts are really important because if you've ingested fat, fat is good food. Look at all of those calories there. But the problem is that fats don't dissolve in water. And the chyme, resulting largely from, gastric juices, juice from the small intestine, it's mostly water. So if we can't break this droplet into smaller pieces, then this will pass through the large intestine and be released from the body as waste. But because we need so many calories to get through a day, that would be horrible, to starve to death on a given day when you ingested enough calories, but you couldn't break that down. So instead, we want to turn these large fat droplets into smaller ones. And that's what bile salts do. Bile salts break large fat droplets into small ones that can then be digested. That's what's referred to as emulsifying a fat. And so thus bile is made in liver lobules. It travels through these bile canaliculi, traveling from the middle towards the exterior. And then the bile then passes through the hepatic ducts into the common bile duct. Now, because we're not constantly ingesting fat, the bile can be stored, so it can back up. And instead of then being released from the common bile duct, it could then pass through the cystic duct into the gallbladder. The gallbladder will then store and concentrate bile. So the gallbladder does not make bile, it simply stores and concentrates it. And so now if you eat a fat-rich meal, not only can bile go from the liver through hepatic ducts to the common bile duct, but also concentrated bile can go from the gallbladder through the cystic duct to the common bile duct. And this will allow us to emulsify the bile in our diet. Now, you may have noticed that the common bile duct joins to the pancreatic duct where it meets the small intestine. So there's this raised region, the hepatopancreatic ampulla, where both the common bile duct emptying bile and the pancreatic duct emptying pancreatic secretions empty into the small intestine. And they are then controlled by one common sphincter. There is a sphincter of the hepatopancreatic ampulla, which then controls both the release of bile from the common bile duct and the pancreatic secretions from the pancreatic duct. So this first region of the small intestine, known as the duodenum or the duodenum, I think its name literally means 12 fingers in Latin, because it's the shortest region of the small intestine, maybe 12 fingers in length. This is a busy region. Not only does the stomach empty here, the pyloric sphincter opens, marking the entrance to the small intestine. But this is also where the hepatopancreatic ampulla empties both the pancreatic secretions and the bile from the liver and gallbladder. Now, the duodenum looks just like the other two sections of the small intestine. All of them look the same on the outside, but they are different. They are, they can get blood from at different regions. So the gastroduodenal arteries can treat the duodenum rather than the other regions of the small intestine. And once again, you can see how busy this is. The stomach empties here, the pancreas and the common bile duct empty here, all right? But histologically, it's different. As you'll see in the next video as well, there are glands, this portion here, these are Brunner's glands that are only found in this section of the small intestine. Also, a great deal of bicarbonate needs to be released because the intestine is basic, so therefore it must neutralize the acids of the stomach. And so the duodenal glands are unique. And so this first section of the small intestine, the shortest section, then is recognized as distinct from the others. Right? So here we have the duodenal glands, Brunner's glands. Once again, they have to neutralize the stomach's acid, because while the enzymes of the stomach, like pepsin, they need to work in an acidic medium. The stomach acid would then denature enzymes like trypsin, which are working in the small intestine. So while the duodenum needs, or while the stomach needs to be acidic, the small intestine is basic and needs to be thus. So when you look at the sections of the intestine under the microscope, look here, the duodenum has these prominent glands secreting digestive enzymes, but then also neutralizing stomach acid. Notice that these Brunner's glands are absent in the next two sections. Notice that the final portion of the small intestine, the ileum, has these large masses of immune cells known as Peyer's patches that are absent in the duodenum or the jejunum. And then notice that the jejunum can be defined by default. It has neither of those. It has neither the Brunner's glands of the duodenum nor the Peyer's patches of the jejunum. So, I'm sorry, the Peyer's patches of the ileum. So here are the Brunner's glands of the first portion of the small intestine, the duodenum, and then, as I had mentioned, there are those prominent Peyer's patches, which are masses of immune cells in the ileum, and the jejunum has neither of those. Okay. So perhaps the most important thing about the small intestine is that it wants to touch your food. All right. This is where most chemical digestion occurs. This is where most nutrients are absorbed from your food into your bloodstream. So one could argue that this is the most important part of the digestive system, the small intestine. It needs to touch your food. Many of the enzymes which are doing the digesting are actually attached to the intestinal cells. So they're part of the brush border. They form these brush border enzymes. So the only way you digest your food is by touching it. And certainly the pumps which then absorb nutrients are in these cells. So you have to touch your food in order to absorb the good stuff. Okay. So you need to touch your food in order to get the calories that get you through the day and the nutrients. And remember, the only way that you're 100 degrees with a big brain and an active lifestyle is if you get an incredible amount of nutrients and calories compared to most vertebrates. Mammals, and particularly primates and humans, are incredibly expensive. You know, just for comparison, a zoo can keep maybe 40 big alligators on the same amount of food as one tiger. Mammals are just expensive, and if you can't pay for the mammalian, physiology, well, then you're going to starve. So we need to touch our food. And when we look at the small intestine, we see that the small intestine has three things which maximize surface area for this touching. First, the mucosa is not, perfectly flat. There are these folds, all right? So there are folds in the intestinal wall. Look at this. So, whoops. So look at this, it goes up, it goes down, it goes up and goes down. So folds in the intestinal wall are going to increase surface area, okay? Secondly, however, notice that the lining isn't flat. Look at all of these cells. The cells of the small intestine, they go up, they go down, they go up, they go down, they go up, they go down. These form finger-like projections known as villi. All right, and these villi then greatly increase the surface area. There are so many more cells touching the nutrients in the diet than otherwise would be the case. And then a third factor is if we were to look in the cells themselves, then you would see that not only are there villi made of many cells, but then there are also microvilli. You'll notice, however, all of the blood vessels here. So obviously, when we digest our food, the nutrients from the lumen of the digestive tract here then get broken into smaller pieces and get into both the blood capillaries of the small intestine, and also there's a lymphatic capillary known as the lacteal. So you'll notice that there are prominent vessels, including a lacteal in the middle there. But here on the surface of these cells, notice that they have microvilli, all right? And so microvilli are now these finger-like extensions on the individual cells of the mucosa themselves. This is where many enzymes are located, and so many of these enzymes are called brush border enzymes. They're on the microvilli, which look like, under the microscope, a brush. So the small intestine touches your food because it has both folds, villi made of many cells, and then microvilli, all of which then increase the surface area of the small intestine, allowing us to digest our food far better and to absorb the nutrients. If we didn't have these folds, villi, and microvilli, we would need maybe about 2,000 times the volume of the small intestine that we already have. So it is here along these microvilli that many of the enzymes which are digesting the food, and certainly the channels and pumps which then absorb the nutrients are located. Okay. The small intestine has a sphincter which serves as the entrance, that is the pyloric sphincter. And then there's a sphincter which is the exit from the small intestine, and thus the entrance of the large intestine. So the ileocecal sphincter then marks the distinction between the ileum, the final portion of the small intestine, and the cecum of the large intestine. So this ileocecal sphincter is then the entrance to the large intestine. Now, while some material can then kind of go into this blind pouch known as the cecum, most material will then go into what's known as the ascending colon, across the transverse colon, down the descending colon to the S-shaped sigmoid colon. So here's the cecum of the monkey, and then, and here in the humans, one can proceed from the ascending colon, transverse colon, descending colon, sigmoid colon, before forming then the rectum. Now, one of the aspects of health which is becoming better appreciated is the number of bacteria which live in the large intestine. And so we have what's known as a microbiome, bacteria which we carry around with us all the time, about 2 pounds of bacteria. And we're beginning to appreciate the degree to which they help us not only with digestion, so digesting things that we can't, and then we absorb some of the products into the bloodstream, but also the ways that they affect health. So the microbiome is very important. The microbiome then has to be managed, and so the large intestine has a lot of immune tissue which manages the microbiome. But that's one of the roles of the cecum, as I'll mention again in the next video. Now here, notice that the cells which produce mucus have been stained blue, because the large intestine is absorbing a bit of water, while about 1 liter of water will reach the large intestine every day. About 9/10 of that is reabsorbed. But then as the material becomes then drier, having it pass would then be more difficult. And so the cells lining the large intestine include many goblet cells which make mucus. And then this mucus helps the passage of the material from the large intestine. Okay. Now, a lot of the digestion which is occurring here is thus anaerobic. Now, if things are metabolized in air, then you produce carbon dioxide, you produce sulfates, you produce nitrates. Swamps often have an odor because their decomposition is not aerobic. So you're making ammonia, NH3, instead of nitrate. you're making hydrogen sulfide, the smell of rotten eggs, instead of sulfate. And so large intestines, to a degree, often smell like swamps because the digestion which is occurring here is anaerobic. We also then have a type of bacteria known as archaea living in our intestines, which are helping to break down some of the things in our diet. They produce methane as a byproduct, and only these archaea bacteria are actually then methanogens. Just getting back to the cecum in a second. You might ask why we have that blind pouch, and it probably serves a number of roles. One of which is that the bacteria of the microbiome, they're important for lots of reasons. And if we were to lose the healthy populations of bacteria, the kinds we like, well, then unfortunately our large intestine could be infected by bacteria that we don't like. So, infections like C. diff often occur after someone takes an antibiotic, which kills all of the healthy bacteria which we like. So many animals have a prominent cecum. All right, so if you look at herbivorous animals, look at that huge cecum, this blind pouch, which houses the populations of bacteria which help to break down foodstuffs, because we don't depend on the breakdown of things like cellulose to the same degree. We humans, we don't have such a prominent cecum, but we nevertheless do possess a small one. And one of the roles that it would play is that if we were to have diarrhea to expel something which we don't feel we would like to keep in our digestive tract, that's a problem because we now also then lose the populations of microbes which we like. And so we have a cecum which would store some of these healthy bacteria. And then after having diarrhea, this could then repopulate the colon with the good bacteria. From the cecum is a piece of lymphatic tissue known as the veriform appendix. If that were to become blocked from, say, perhaps a solid piece of material, then that could cause the appendix to become inflamed and burst. And that would be a problem because there are trillions of microbes per gram of our microbiome. All right, so the large intestine has the largest accumulation of bacteria in the human body. If we develop a hole here, the amount of bacteria which would be entering the pelvic body cavity would be enormous, and thus we could have, the degree of infection would actually then be life-threatening. And so the bursting of an appendix is, a very serious threat, and thus pain in the lower right quadrant of the abdomen must always be taken seriously, because if an appendix is close to rupture, then a patient could go from a normal day to a life-threatening condition in a very short period of time. Okay. So the first portion of this chapter summarizes the anatomy of the digestive system. The next portion will then go through the physiology of it and digestion.